Hello friends, today we are going to study in detail about process of fertilization from the chapter 2 Reproduction, lower and higher animals. Now let's see about the fertilization. First, definition. The process which involves fusion of male and female gametes resulting in the formation of diploid zygote. It is called as the fertilization. So, in a sexual reproduction, primary involves formation and fusion of gamete. In the human, the process of fertilization is internal and usually takes place in the ampulla of fallopian or uterine tube. See that in the diagram I have shown that ampulla and how that sperm comes in contact with the ovum. So here this fertilized egg or will develop into an embryo and the process occurs within the uterus. That's why it is known as the internal fertilization. Now let's see about the mechanism of fertilization. In the mechanism of fertilization, there are the different events occurs such as the first one, movement of sperms towards egg, entry of sperm into the egg and activation of sperms. Now what happened during this fertilization? That semen released during ejaculation has sperms and some secretions. The coagulated semen is now undergoes liquefaction and sperm become active. The mechanism of fertilization is as follows. It involves capacitation of sperms reaching the vagina. Here as many as 50% are demotilized, broken, destroyed. The remaining sperms undergo capacitation. This process requires 5 to 6 hours. A chromosome membrane becomes thin. Calcium enters the sperm and their tail begins to show rapid whiplash movement. As a result of capacitation, sperms become extra active and begin to start moving upwards from vagina to uterus and into the oviduct. The prostaglandin activate the sperms. The vestibular secretion of sperm of female also enhance sperm's motility. The sperm swims at an average speed of 1.5 to 3.5 mm per minute. Sperms reach up to ampulla as a result of their own swimming and partly by contraction of uterus and fallopian tubes stimulated by oxytocin of female. After capacitation, the sperm may reach ampulla within 5 minutes. Sperms can remain viable for 24 to 48 hours. This ovum for about 24 hours. Next event which occurs in the fertilization process, that is the next event during the fertilization, that is the entry of sperm into the egg. Out of 200 to 400 million sperms, only few hundred manage to reach the ampulla. Though many sperms reach the ampulla, but only a single sperm fertilizes the ovum. A sperm, after reaching the egg or ovum, comes to lie against it. Its acrosome releases lysine, high hyaluronidase, and corona penetrating enzymes. They separate and dissolve the cells of corona radiata, so the sperm head passes through the zona pellucida of the egg. The zona pellucida has fertilizing receptor enzyme and the fertilizing binds to specific acid protein, that is antifertilizing of sperms. It brings about attraction of sperms to the egg to enhance fertilization. Fertilizing and antifertilizing interaction is species specific, thus the fertilizing antifertilizing reaction is also called compatibility reaction. See that in the diagram it is already shown how that sperms comes in contact with the ovum. Now the next event which occurs in this fertilization, that is acrosome reaction. As the sperm head touches the zona pellucida in the animal pole region, its acrosome covering ruptures to release lytic enzyme, acrosine or zona lysine. They act on the zona pellucida at the point of contact. This causes egg reaction. A small fertilization cone or cone of reception is formed on the egg membrane. The sperm head comes in contact with this cone. It results in the production of weak wave of depolarization. Plasma membrane of both cells dissolve at the point of contact. The sperm nucleus and the centrioles enter the egg while the other part remains outside. As soon as the sperm head touches the vitelline membrane, a cortical reaction gets activated, changing the vitelline membrane into the fertilization membrane by deactivating the sperm receptor of zona pellucida. A distinct perivitelline space is created around the fertilization membrane. This prevents any further entry of other sperm into the egg, that is, polyspermy is avoided. Next event, that is activation. The ovum before fertilization was at metaphase second stage. With a contact of sperm head to vitelline membrane of egg, it gets activated to resume and complete its meiosis second. With this, it gives out the second polar body. The vesicle organizes into female pronucleus. At this stage, it is the true ovum or egg. Then, fusion of egg and sperm. The coverings of male and female pronuclei degenerate, allowing the chromosomal pairing. This results in the formation of synkaryon by the process called syngamy or karyogamy. The zygote is formed. The proximal centriole received from the sperm helps in the formation of synkaryon spindle and cleavage of cell into two blastomeres. Now next see fertilization. Secondary oocyte completes the process oogenesis and is transformed into mature ovum. The diploid chromosome number is restored in the zygote by the process of syngamy. The ovum lacks the centriole necessary for further division are received from the sperm during fertilization. Next significance, during fertilization, various occurs which are significant to ovulation. And like this, the fertilization is very important to sex determination of offspring. Like this, in this topic we have seen about the fertilization in detail.